button. So once again, now as a recorded version, a warm welcome in this Eco and Bioart Lab webinar. The Eco and Bioart Lab is a research group and also a kind of research platform that really connects artists, artistic practitioners, artistic researchers, and researchers who engage with art and the environment, broadly speaking. This is also a platform that really connects people from within the academia with those outside, with various institutions and collaborators, among them Bioart Society, the members of which are also present here in this room, which I'm very happy to see, and I'm saying hello, of course, to all of you. You know who you are. And today we are actually meeting for a very special occasion, and this is the webinar Sensory Foraging, the Eros of Olfaction and Multi-Species Metabolisms, with Dr. Tarsh Bates, artist and researcher from Umeå University, and with Professor Emerita Nina Lykke, based or affiliated with Linköping University, but also with Odorants University in Denmark. And we'll have this exciting session with a longer presentation and a kind of response from Nina's side and a dialogue with also space for all of you to engage in. And before I kind of get to those, so to speak, rules, housekeeping rules, or housekeeping information, I'm just going to say that Tarsh Bates has a PhD from Symbiotica at the University of Western Australia, and they have worked as a pizza delivery driver, a fruit and vegetable stacker, a toilet paper packer, a compost researcher, a honeybee ejaculator, a gallery invigilator, a raspberry picker, an academic and editor, a bookkeeper, a car detailer, and a life drawing model. So there is a richness beautiful to that. And of course, they are currently a postdoctoral research fellow in design and molecular biology at Umeå University in Sweden, in the north of Sweden. And of course, Tarsh is enamored with Candida albicans. And I also would like to say a few words about Nina Lykke, who is the respondent to Tarsh's talk today. Nina is a poet, philosopher, and professor emerita of gender studies at Linköping University, but also an adjunct professor at Aarhus University. She has participated in the building of feminist studies in Scandinavia and Europe more broadly since the 1970s. And she is also the co-founder of the International Network for Queer Deaf Studies. And Nina's monographs include Cosmodolphins, published in 2000, Feminist Studies from 2010, and very recent book, exciting book, Vibrant Deaf from 2022. So when it comes to housekeeping rules, and I'm kind of, still admitting here people as we speak, we'll have now kind of around 35 minutes for Tarsh's talk, and then we'll move to the kind of, around 10-minute response and the kind of dialogue between both our speakers and researchers today. After that, I will invite you for a, kind of a little stretch break or, comfort break, and then we reconvene for a kind of joint Q&A and broader discussion. Right, so I think that, I kind of did the sort of opening of the session much faster than I thought, which also means that I will, with a great pleasure, pass the baton to you, Tarsh. And yes. So let's try to share the screen. And to all of you in the room, I mean, we of course tested that, but I mean, let's see how it goes this time. It always is the situation that you test, it goes perfectly well in the test, and when you have the main kind of right, then things have their own life, so to speak. So let's see. Fabulous.
Excellent. Let me just make sure. Yep, perfect. Thank you so much, Marietta. Thank you, Nina. And thank you all for whoever's in the ether. I really appreciate you coming. I hope that this does something. Let's see. Okay. I am a non-Indigenous Australian. I was born in Manunjalibo Desert, Yugambeh, Queensland, grew up in Matamata, Aotearoa, New Zealand, and have lived for the last 35 years in Boorloo, Perth, Noongar Boodja, southwestern Australia. I pay my respects to the Whadjuk people of the Noongar Nation and the Sami people of Umeå Sapmi, the lands on which I live and practice, their elders past, present, and emerging, and their continuing cultural, spiritual, and educational practices. I am formed from the exhalation of cyanobacteria. And millennia of evolution. My body seethes and pulses with hundreds of other species, fashioned and transfigured by tiny lives and deaths, host to a thriving ecology. We are in relentless reorientation, sensitive to pH, moisture, temperature, and nutrients, tentatively traversing the affordances of each other's bodies. Our bodies are animated by chemical transmissions and constant reproduction, transfigured by sensation. As a scavenger host, I am responsible for my ecology, to care for and about it. I forage knowledge, materials, ideas, tools, and hunt transient alliances. I transmogrify ingredients into evocative victuals, sampling and nibbling, testing and sipping, kneading and folding, assembling and serving. I offer a trans-species degustation, companionable portions for consideration and transformation. I transmute the human body into a more than human ecology, a site of sustenance for non-human life, food and shelter, reproduction and kinship. I am one of a handful of humans on the planet who is in love with Candida albicans, a yeast, one species of the hundreds that live in the ecosystem of the human body. Candida is a single-celled fungi, a yeast. It is thought to have not evolved in any other organism. It is both commensal and an opportunistic pathogen and occupies almost every ecological niche in the Homo holobiont. We have co-evolved. Human bodies are the ecological niches of Candida albicans. We are their hosts, providing them with food and shelter. Holobionts are coagulations of hosts and many other species living in or around them, an ecology in constant formation and deformation through symbiotic metabolisms. Holobionts include the host, virome, microbiome, and any other organisms which contribute in some way to the functioning of the whole. This functioning as a whole is symbiosis, a term coined by Beth Dempster to refer to collectively producing systems that do not have self-defined spatial or temporal boundaries, are evolutionary and are characterized by continuing complex relations among system components, she says. For Haraway, symbiosis is making with. Anna Tsing insists that this making with is never limited to humans, since all organisms make ecological living spaces, altering earth, air, and water, thereby producing workable living arrangements, whereby each organism changes everyone's world. Marie Catherine Petersman argues that this life-making process unfolds through the interactive relating of holobionts, where interaction signifies the mutual constitution of entangled human, non-human agencies. The Homo holobiont is a queer ecology, a complex and diverse entanglement of relationships between Homo sapiens, Candida, other microbial species, culture, and technology. Queer ecologies disrupt the mutually constitutive apparatuses of nature and sexuality, and reconfigure the entanglements of biology, sex, sexuality, intimacy, affiliation, geography, geology, ecology, culture, and technology. This figuration enables me to explore how Homo holobiont relationships are constituted and who gets to be at the table when and where. The human body is a profligate beat of more than human sexualities, where human and microbial cells, including Candida, replicate, procreate, and propagate. Matthew Gandhi suggests that a queer reading of space reveals a distributed agency of desire that extends beyond individual or even multiple human bodies to incorporate non-human nature, inanimate objects, surfaces, and smells. The sexual body is no longer one human subject, he says, but an array of different elements that dispels any attachment to residual humanism. However, as Kath Brown argues, queer is not a simplistically appropriated identity category, but a fluid set of possibilities and contestations. Queer is the pleasures and struggles of the everyday, the iterative unconscious generation and regeneration of identity, the transformation and modification of flesh, the grief and trauma of alienation and toxicity, the deep temporality of plastic and fossil fuels, the biopolitics and eros of desire, and the commitments and alliances of flourishing. Heather Davis has argued that environmental activism is futuristic and fundamentally heteronormative, since futurity implies reproduction. We are urged to save the environment for our children, or even to consider 7 generations ahead, as Indigenous cultures do. Lee Edelman has famously argued that since queers don't reproduce, we have no obligation to the future. According to this logic, future-oriented environmentalism has no purchase for queers. Why should we care? However, as Neil Ahuja argues, queer humans are not immune from the parasitic exploitation of more than human sexualities and are complicit in the microbiopolitics of reproduction and extinction, as he says, where racial divisions of climate emerge in the intimate scales of contact between human social forms and ecologies of production and waste. Davis provides a compelling argument for reconsidering queer space-time matter, not as anti-futuristic, but as an always already present. If we have only the now, what we do and who we do it to and with matters here now. The motivation for changing our behavior must hinge on more than reproduction, more than lineage, more than the future, since we are in it now. The space-time matters scales of the Anthropocene are horrifying, urgent, and overwhelming. As Haraway argues, we are all responsible to and for shaping conditions for multi-species flourishing in the face of terrible histories, and sometimes joyful histories too. But we are not all responsible in the same ways. The differences matter in ecologies, economies, species, lives. Homo holobionts are as much a part of the Anthropocene as any other. Life at all scales depends on biodiverse metabolisms. In untangling and reconfiguring the small bodies and intimate environments, as Ahuja says, of Homo holobionts, I urge us to reorient to the responsibilities of our own ecologies. Therefore, I orient a queer ecological sensibility towards the microbiopolitics of Homo holobionts. Microbiopolitics, as theorized by Heather Paxson, draws from Michel Foucault's biopolitics, and Bruno Latour's analysis of pasteurism describes the creation of categories of non-human biological agents, the anthropocentric evaluation of such agents, and the elaboration of appropriate human behaviors given their entanglement with microbes engaged in infection, inoculation, and digestion. Ahuja explicitly intersects microbiopolitics and queer when he argues that queering in this sense emerges by tracing an effective materiality that interrupts anthropocentric body logics and time and space-time continuums rather than a sovereign stance of negation in relation to law, including the law of compulsory reproduction. It is an inquiry into the unrealized life worlds that form the background of the everyday, This requires thinking askance the human and thinking death, animality, and vulnerability in an age of many extinctions. Heather Paxson insists that microbiopolitics is one way to frame questions of food ethics and governance. I'm interested in forming heterotopic alliances that defend the marginal and interstitial spaces of the Homo holobiont and teach us how to eat better together. Luce Irigary's conception of eros as an ethical encounter firmly grounds Emmanuel Levinas's ethics of difference in embodied intimacy, in the caress. In the fecundity of the caress, Irigray configures eros as a rebirth of self and other. She rejects Levinas's eros as an antisocial encounter between two that leaves the other as other and procreates a third individual. Irigray argues that we are at least three, and the caress of at least three generates difference, not individuality. The caress opens Levinas's radical alterity from the visuality of the face-to-face relation to the voiceless and the sensual. Surfaces become membranes that enable fluid exchanges, reciprocities that trouble self. Christopher Cahoon argues that Irigray's caress is inherently reciprocal, a call to coexist. In caressing, touch renews openness to each other in a recirculation of sensation that generates a commensal pleasure, a feeling otherwise. Eros is a fecundity that proliferates radical difference, not a variant of the same, not procreation. It is highly specific, converging on the loci of the caress, and so cannot totalize the other. The caress is spatially and temporally ephemeral, quanta of difference that cannot represent the world, cannot be universalized. The caress of eros is ephemeral, fleeting, exciting, and invigorating. It is the stroke, the tickle that stimulates laughter if only performed by another, quickly, gently, unpredictably. It is the essential affective desire for interconnection and interaction that exists simultaneously and paradoxically alongside perception of difference and desire for autonomy. Bodies subject to Irigrayan eros exist as potential energy in states of imminent becoming with. Matter is a generative opportunity where membranes are zones and processes of differentiation, and differentiation spawns difference. Tim Morton extends Irigray's eros to all beings and ecologies, arguing that ecologies and their components can never be categorically measured as they are in constant formation and generation. Eros itself offers a queer ecological ethics as creative fluid work that regenerates subjects. This work is not comforting or comfortable. The caress is friction that activates, irritating and unsettling. As Karen Barad describes, all we ever really feel is the electromagnetic force, not the other whose touch we seek. Electrons which lie at the farthest reaches of an atom, hinting at its perimeter, cannot bear direct contact. Electromagnetic repulsion, negatively charged particles communicating at a distance, push each other away. That is the tale physics usually tells about touching, repulsion at the core of attraction. The caress of eros is electromagnetic repulsion, essential, interstitial, etc., and always already becoming with. It is impatient and restless in constant flux, the most extreme experience of sensation, Irigray says. Irigray argues that if touch remains always invisible as such, some parts of our body that are particularly concerned by eros are also invisible. It is especially the case for mucous membranes, which are the most sensitive parts affected by an erotic awakening and touching. So eros, by definition, extends to the more than human, to microbes who dwell in those sensitive mucosal membranes, invisibly touching, and whose entire membrane is a sensor affected by erotic awakenings and touchings. Although laboratory living tends to be a solo affair for microbes, the Homo holobiont is a crowded place. Encounters with other members of the microbiota, host cells and abiotic materials, including nutrients, toxins, steroids, antibiotics, biomedical devices, and volatile chemicals are inevitable. These encounters are common and extremely complex. In addition to their engagements with host cells, microbes compete with other microorganisms for niches, adhesion sites, and nutrients, and deal with toxins and metabolic byproducts of their neighbors. They also cooperate and cohabit with other species, share host niches and nutrients, form protective biofilms, and exchange metabolic byproducts. Inter-kingdom interactions are affected by conditions of the host niche, species composition, and the presence of antibiotics. These complex interactions and encounters are mediated by chemical signaling and surface sensitivity. This is an environment where contact zones are physical, vision is irrelevant, touch is all. Bodies brush against each other and their surrounds, and communications are visceral. Chemical signals excreted and received by cell surface moieties. The microbes literally grasp their environments. Communities form and dissolve as communication is ingested, digested, and excreted. Microbes are intensely social, communicating and cooperating to perform a wide range of multicellular activities, such as dispersal, nutrient acquisition, protection, biofilm formation, and developmental quorum sensing. These social behaviors are mediated by intercellular chemical and physical signaling and are the rule, not the exception. The production of these signals, so-called common goods and services, for transport, protection, and nutrition is fundamental to microbial survival. This is an erotic multi-species orgy of metabolisms, microbial and human cells engulfing, engorging, erecting, ejecting, roiling and spoiling, caressing and converting nutrients, hormones, proteins, minerals, steroids, plastics, vitamins, sugars, caffeine, smells into energy and matter. Haraway points out that our intimacy with our tiny messmates literally defines our survival, and for Derrida, eating well is never done alone. Drawing from Hannah Landecker's understanding of food as a conditioning environment that affects genes and physiology, Myra Hurd argues that metabolism precedes reproduction and is the force by which life continues to produce itself. It relentlessly recycles materials, providing energy for life and non-life. Metabolism, she suggests, is an entangling force as it defines a relationship between non-discrete entities, themselves already entangled. Through metabolism, environment becomes organism, and organism becomes environment, an inorganic activity through which cells, organisms, life, and non-life maintains, develops, and responds. Similarly, Landecker argues that relational biology is increasingly concerned less with the qualities that compose a given bounded biological entity and more with a biology of the in-between. The human animal body is sustained not through the food it currently eats, but by the food it has previously ingested that has become part of the body itself. As an in-between, metabolism is therefore fundamentally queer. Landecker further asserts that we cannot help but ingest, and in the act of digestion are drawn into the social, technical, and political networks of food production, regulation, and consumption. We are what we eat, but also what our parents and our grandparents ate. Hurd suggests that human cultures are intensely defined through aspects of metabolism, including taste, ritual, intolerances, allergies, and so on. I add olfaction to Hurd's list of metabolic aspects that affect human culture and argue that olfaction is a crucial and neglected aspect of metabolism. Each day we breathe about 23,040 times and move around 438 cubic feet of air. It takes us about 5 seconds to breathe, 2 seconds to inhale, and 3 seconds to exhale. And that time, molecules of odor flood through our systems, inhaling and exhaling. We smell odors. Smells coat us, swirl around us, enter our bodies, emanate from us. We live in a constant wash of them. Still, when we try to describe a smell, words fail us like the fabrications they are, Diane Ackerman says. Caress your neck gently. Smell your fingers. Caress someone else's neck. Let them smell your fingers. Olfaction is the sensation experienced by the caress of volatile organic compounds on the membranes of cells. Gas phase metabolites such as microbial VOCs diffuse across cell membranes much more efficiently than fluid or solid metabolites. The Homo holobiont is awash in microbial VOCs, an erotic inter- and intra-species communication, a metabolic sensibility that traverses space-time matter. Odorants, the chemicals that produce smells, are fundamental within metabolic processes, ingested, digested, excreted through the living, the non-living, and the semi-living. Current metabolic studies tend to focus on non-volatiles. However, MVOCs are overall mediators of interactions in microbial metabolic systems. Within the Homo holobiont focuses on diagnosis of illness. Microbial VOCs can be tracked in real time to track microbial activity. I'm just going to skip a few of these because we are out of time. These are other artworks related to olfaction. And we can talk about them later if we want to. Okay. Humans are always already manipulating the evolution of others and manipulated in turn. However, we are reorienting to an unprecedented scale of manipulation and industrialization of yeasts and other microbes for consumption, certainly, but increasingly we're exploiting their sexual and reproductive lives to produce industrial and commercial enzymes, pharmaceuticals, biofuels, proteins, flavors, and fragrances. Monica Bakker digs into the deep temporal-materiality of metabolism when she examines the biochemical flows between geology and biology. Following Karen Barrad's claim that matter is never settled matter, she argues that the very materiality of metabolism offers an infinity of possibilities of its provisional unfoldings, evoking a humbling and empowering experience of both infinity and immortality. Through our participation in metabolic networks, which include photosynthetic life and mineral species, our bodies, she says, reach towards the sun and the mineral formation of the earth and other terrestrial planets and moons, or what Rosie Bradotti describes as becoming earth and Catherine Yusof as geological life. Bakker argues that the anthropogenic rearticulation of metabolisms using technoscientific tools such as synthetic biology and nanotechnology is shaping future environments that will most likely sustain resilient life forms that are other than human, an infinite transformation of matter, allowing one to think of the future of the human species in other than catastrophic or alarmist terms. Every here now is a moment to be responsible, sensible. We decide here now how we want to eat at the table. Do we plead austerity, rationing, and dispensing morsels based on capricious judgments, allowing some to gorge and others to starve? Or do we invite everyone to a generous, fragrant table of mutual flourishing, an orgy of queer kind, and deal with the hangovers and food poisoning, the fisticuffs and arguments, the STIs and accidental progeny, the detritus and the stench? Close your eyes. Breathe in 400 milliliters of air. Breathe out 400 milliliters of air. Breathe in 400 milliliters of air. Feel the zephyr of volatile organic chemicals from plants, soil, fungi, water, food, shit, the dust of stars flow over the surfaces of your nasal cavity and caress your olfactory bulb. Count the microbes settling in the niches of your pharynx, larynx, trachea, and lungs, feasting on the VOCs you have inhaled. Breathe out 400 milliliters of air. Trace the marks left by the microbial VOCs on your trachea, larynx, pharynx, nasal cavity. Mold the shapes of the molecules that swirl over your lips in a moist, volatile stream. Release them from the membranes of your body and map their caresses of the person next to you, of plants, animals, fungi, microbes, soil, lakes, and oceans. Breathe in 400 milliliters of air. Breathe out 400 milliliters of air. Open your eyes. Thank you.
Thank you so much. Fantastic. Okay. I think then we can open the floor or pass the baton to Nina. That's why I kind of keep you, still the two of you as kind of like spotlighted in this view for everyone. For Nina's engagement with Tarsh's talk, and then we'll have a bit of a dialogue as well. So Nina, the floor is yours.
Okay, thanks so much for an amazing lecture and slideshow, and I was actually very, very happy to have been able to read it on beforehand to engage with it. And I have been looking a lot forward to that, because both the talk and your slides gave me a lot of food for thought. I was a bit sorry that you left out all the artworks on olfaction, but as you said, perhaps, yeah, well, they might come up in the discussion. You might refer back to them or whatever. But I mean, your lecture gave me a lot of food for thought, and digesting it made me think about many things to be in the metaphorical language of metabolism. And I'll share some of these thoughts here. First, I would like to compare notes with you on relationship with strange critters. Being myself a queer lover of strange, minute creatures, in my case, the single-cell algae diatoms, which fill waterways all over the planet and produce 25 to 30% of the planet's oxygen annually, I can definitely follow you in your love for the microorganism Candida albicans, a truly, truly interesting creature whom I did not know with my conscious mind on beforehand, even though it's probably all over my body and has been there since the day I was born. My work with diatoms is, among others, directed by my feeling that it's truly important and urgently needed in our present times of Anthropocene necropolitics and mass extinctions, that we humans take a lot of measures to make sure to undo our arrogant human-centeredness and belief in human exceptionalism. One of these measures, which I myself have tried to take seriously, inspired by, among others, Donna Haraway's work on kinship making, the make kin, not babies slogan, is to establish cross-species companionship with diatoms, in particular the ones that exist in the waters where my beloved queer masculine life partners' ashes are scattered. So I have a very personal relationship to these diatoms. While reading and listening to your lecture, I felt invited to speculate in depth about your companionship with Candida albicans, a companionship which I somehow saw as a bit parallel, also very different, but some parallels I saw, at least, to the one I have established with the diatoms of Limfjorden. Limfjorden is the place where my partner's ashes are scattered. And of course, a companionship which is also parallel to the one Donna Haraway speaks about when she takes companion species beyond the relation to her dogs, which of course is interesting, but I think even more interesting when she talks about a companionship as, I quote, a pointer to an ongoing becoming will. And I do think that's very much ties into what you have been talking about in When Species. She talked about that. I mean, it's a quote from When Species Meet. That is to a shared process of mutual co-becoming across species borders, which Haraway also exemplifies with her relationship with her gut microbiome. So I think there's plenty of resonance here. Taking Haraway's companionship concept further, I've come to consider my own relation to the diatoms as a queer love relationship and a companionship. And I'm also, within this bigger framework, trying to establish an eco-advocacy for them. Along the lines of eco-advocacy, and among others, trying to make it known that when seascapes die, which they do very much, due to oxygen depletion, which is wrongly blamed on algae such as diatoms and cyanobacteria and others, it's of course human-induced, among others, agricultural discharges and nitrogen and phosphates and other things that's actually to blame. But when seascapes die, it's not only multicellular organisms such as fish, mussels, seagrass, etc., who die, which is what is in the media, what's happening in catastrophes, etc., but the microalgae die too. I think there is an urgent eco-ethical need for this kind of foregrounding our human symbiotic interdependence with the world of microcritters, from diatoms to Candida albicans in these times of Anthropocene necropolitics, where stupid human exceptionalism is still the predominant way in which humans consciously approach the world, and especially, I mean, the world at large, of course, but especially the world of seemingly inferior, invisible, unimportant microcritters, such as diatoms or Candida. And I wonder whether or not you would recognize this kind of move in between queer love and eco-advocacy in your relationship with Candida albicans. So that's somehow my first comment. Then I have a comment question around the notion of futurities and your critical approach to Heather Davis, Edelman, etc. I mean, this discussion about futurities and queer and queer ecologies. To me, it's eco-ethically important to become aware of all the many dimensions within which we constantly interact symbiotically with the world, and to really do this in an in-depth corpo-affective way. And I think it's helpful to concentrate deeply on one critter at a time, which you do in your case, Candida albicans, in my case, the diatoms. I think many new ecologically and personally important lessons can be learned from the establishing of such what I would call in-depth compassionate companionships. Along similar lines, I have recently been collaborating with a Dutch artist, Lykke, who explores their queer love relationship with cyanobacteria. And Lykke and me actually published our collaboration in the Dutch queer art magazine, Forum Plus, in 2022. And it was actually through Lykke and also a bit Elizabeth Covinelli that I learned about the ways in which all the beings, including humans, who today are dependent on oxygen, are an afterbreath of the ways in which cyanobacteria started to fill the air with oxygen eons and eons and eons ago. The great oxygenation event to which you also referred briefly in the beginning of your talk, to illustrate this kind of totally embeddedness of humans and this world of microorganisms. Against the background of my queer love and feeling of deep kinship and desire to explore all possible dimensions of my interactions with the diatoms, I think that Edelman's rejection of futurity as just heteronormative, focused on heteronormative reproduction and therefore outside of a queer or uninteresting in a queer horizon, is in a way much too narrow and much too human-centered. And I guess we are somehow on the same page in terms of finding this Edelman approach to futurity too narrow. Still, I wanted to hear more about your approach to futurity, and of course Edelman if you want, but I think futurity more overall. I guess that we are somehow on the same page in terms of transgressing Edelman, though I also guess that we perhaps differ a bit in terms of our answers to the question what's wrong with Edelman's approach. And I'll come out my answer a bit very briefly, and then we can compare notes. I do think that futures are not by necessity inscribed in hetero-reproductive terms. as Illiman claims. That's what I think is human-centered. As an alternative, I would go with Haraway's "making kin, not babies" slogan and claim that I want to care about the futures of my queer kin in this Harawayan sense. That is, my queer kin understood as the symbiotic networks of entangled living and dying critters, diatoms, Candida albicans, and others. And I hope that these networks will continue in a vibrantly ongoing way, rather than being broken by what Deborah Bird Rose called enocide or double death. Anthropocene mass extinction without possible for any kind of regeneration. This approach to futurity is related not only to ecoethics per se, along the lines of Deborah Bird Rose, even though I like the word very much, but also to my thinking around death, which I do not consider as an endpoint, but rather as a material transition to other forms of vibrantly material and entangled becoming and decomposing. I care about modes of contributing to making death for all creatures vibrant rather than making it into extinction and double death, and do that in collaboration with my queer kin, all these critters. However, I can also follow you on your concentration on the queer, what you call here now, rather than on the future in an abstract sense. That is, your concentration on the exploration of the queerly loving cohabitation and interaction with Candida albicans in the temporal mode of the here now. In my work, I've argued for connection to the non-chrononormative time of Aeon, A-I-O-N, along the lines of Deleuze. And for me, Deleuze's description of the time of Aeon encompasses not only the time of life, but also the time of death. And that's what I think is important. And the being with the dead in an ongoing relation, which I really think is really important to integrate in these discussions. My connection to temporalities, which are not circumscribed by an individual lifetime, became very strong and passionate after my partner's death. As a queer femme mourning I lost interest in the future, focusing instead, and I couldn't use the queer here and now moment either. I actually needed the non-chrononormative time of iron and the multi-directional instant, which I've reflected on. It transgresses borders between the world of the living and that of the dead. And I wonder what you think about this integrating the life-death continuum in relation to the question of here now, futurity, cohabitation with the world, basically, the more than human world. That's my second question. Then I have a third one. Do I have time for that? Okay, so the third one is on olfaction and as an ethico-political tool. And related very much to the part, the last part, or the second part of your talk, where you concretely dig into olfaction and metabolism and touch and caress along the lines of Lykke. I read your reflections around metabolism and olfaction as a way of exploring your relation to Candida albicans. And for example, when you experiment with it as yeast to be used in baguette baking, I enjoyed that really a lot. As I understand, this part of your artistic creation experiment, the baguette baking thing, using Candida albicans as yeast, I somehow thought that it's a way of, this part of it is a way of exploring, or perhaps better, provoking the mode in which we humans consider a lot of the micro critters that we live symbiotically together with, and that are truly important for us, and we for them, of course, but also they for us, that we consider them as abject and contagious beings rather than as companions. I also found the artworks you showed exploring olfaction extremely interesting, but they were not in your talk now, but only in the in the version I read, so I'll not comment too much on that. But I do think that olfaction is a really, really, really interesting, but very much overlooked and also very, very colonized sense, repressed among others by what you, with a wonderful word, called deodorization, which was not in the talk now either, but I really think that we need to undo and decolonize olfaction and well, and undo the deodorization. For sure, lots of human and non-human animals use olfaction as a really, really central sense, where we as modern humans try to avoid all bad smells. I love to see my cat smelling her way through new things that I might have brought into our shared living space before she accepts their presence there. I also came to think of my friend, the Chilean curator Camila Marambio, who together with the US-based artist Christy Gast, as well as Chilean biologist Georgia Grals and Derek Cocoran, did an art project with beavers in Tierra del Fuego, Chile, called Dear Enemy. In the project, they tried to communicate with beavers through smells. Beavers are considered to be so-called invasive species in Tierra del Fuego. Invasive means that humans brought the beavers to the island in the mid-20th century to hunt them for fur. The beavers then tried to make the best of the situation in their unwanted, unchosen new environment, and they did it very well from a beaver point of view. But the humans then started to persecute the beavers because their behavior is disruptive to fragile Arctic ecosystems. So the conservationists even started to plan for a final eradication of all beavers in Tierra del Fuego with drones and everything. However, Camila's, my friend's group, tried to intervene, arguing that it was better to try to communicate with the beavers than eradicate them. And to do so, they came to think of using smells and got a grant to develop beaver sense at Los Angeles Institute of Art and Olfaction. This art science project called Dear Enemy that aimed communicating with the beavers through smells seemed actually to have made the conservation scientists who were invited to participate in the project somehow changed their minds about the beavers and somehow at least put the eradication project on hold, which I think is very interesting. So to make a very long story short, I think that the focus on olfaction is truly important in terms of approaching and thinking with alien encounters with the modern human world differently, since smell is such a key communication device in so much of the modern human world. And your work inspired me to start thinking about olfaction as a truly ethico-politically important tool to establish such much needed more than human communication. And if it could even persuade the conservation scientists and a lot of other scientists, I think it would be even better. I hope you will compare notes with me on this as well. But I have also a very concrete last question, because there is a thing that I did not quite understand, because what is the link concretely between olfaction and Candida? How does Candida smell? I would be very curious if you could say a bit more about that. Thank you.
Thank you so much, Nina. And I mean, I should say that, we are actually very optimistic. We kind of, I think, some minutes ahead of the schedule, which means that, please feel free, Tarsh, to engage with Nina's comments. And if you want to have like a little dialogue, keep on going. And at some point, I will announce the break. But I mean, please, I think that we can, easily take 10 minutes or more even for a dialogue right now.
Wow, how exciting. Thank you so much, Nina. Fantastic questions, and so wonderful to hear your comments about each part, and so much. Okay, so let me think about. Okay, so if we go back to your first question about queer love and eco advocacy, can I ask you how you are enacting eco advocacy with your diatoms?
I'm somehow, I'm engaged in the question of, politically, activistically with groups who in Denmark who are actually struggling because our coastal waters are actually in a state of, serious dying. And the point is that and it's even become so serious so that even politicians in parliament have started to talk about it. But of course, the big obstacle is agricultural resistance, and they have a lot of influence on the government, etc., etc. But the point is that no one talks about that the microorganisms die. Everyone talks about the fish and the seagrass and the mussels and the oysters, etc., which of course is a terrible catastrophe. But the microorganisms are somehow considered to be complicit with the with the ones causing death. And I'm really trying to make that argument in public that the microorganisms die too, and that's fatal.
Yeah.
So that would be part of it, but yeah.
Yeah, I think that I'm not I guess mine is my eco advocacy, I suppose, is a much more less activist one, I suppose, and that the artworks and the conversations that I have are I suppose a more of a consciousness raising activity, maybe at this point. The olfaction project is very new and I'm still working through it, and it's actually part of it's sort of my activity in it has started as part of my postdoc in Umeå, and I will be working in Australia as well. And as part of that, I'm looking at workshops that explore the role of olfaction and microbial olfaction in ecological change. And so I think that will be looking, I guess, at the moment on a more individual, human and personal level about grief and loss and change and how smell works in those systems, and to ask people to think about the other creatures and particularly the microbes that are living in those systems or dying in those systems. So I think that maybe a more activist advocacy will come out of that potentially. I'm hoping as well.
But when I said eco advocacy, I think it can be tons of different things. I mean, it doesn't necessarily have to
absolutely to
walk in the streets to say Candida albicans, everyone be aware of Candida. but
yes.
So I do think that this consciousness raising and this workshop that you showed, using the baguettes and offering people the baguettes, and the baguettes being made by this yeast, which people might find a bit of feel objected about. I think that's kind of what I would consider eco activism.
Yeah, sure. I think that I'm also in part of the olfaction work and the postdoc, very interested in microbial labor politics as well. And so with a colleague, I'm writing a paper about microbial labor. And she actually has an artwork that looks at how to support microbes to go on strike, which I think is very lovely in a very union-oriented country like Sweden. I just love that idea. So she and I are going to be writing about that project. And
Nice.
yeah, I think that another part, I think that it's not enough just to love. Or for me, love means advocacy. I don't see them as separate. Yeah, they're entangled. I guess that's the short.
Yeah, short. I mean, I would agree very much, and I do think that, I mean, what I would call artivism is, I memean, is somehow also based on this love and compassion. And
I think it's Ira Gray who talks about love as a verb. And that was very powerful for me, and I found out about it many years ago, and it's very, very powerful for me. And so trying to treat the things that you love better than strangers is a really important part of the way I live in the world. But coming back to your idea of the making kin, not babies, is who do we include as kin? And so the stranger isn't necessarily somebody that we know, but is kin. And we decide who kin is, right?
And I do think, I mean, I think that Haraway's point in staying with the trouble is in a way that which I'm that I mean that I mean all the world is kin, all these, I mean, all these, because if symbiosis is what somehow constructs, I mean, the world is made in symbiosis, so we are somehow in that sense all in it together as
absolutely.
So therefore basically everything is in that sense kin, and I do think that's I got the idea of actually thinking about taking one species out of that whole, not to separate it as, okay, this is my kin and that's the only thing I care for, but more in also inspired by Haraway, the ways in which she in the Camille stories, in the second part of Staying with the Trouble, actually could somehow in a fictional way construct these links between that all the human families are taking, I mean, the Camille's are taking the monarch butterflies, because it's difficult to, I think, to care. I mean, you can care in the abstract, but you cannot do eco-advocacy, and you cannot go in depth with finding out about a lot of your relationships to everything. So therefore, this somehow that, I mean, I select the diatoms, you select the candidate applicants, someone else might select something else, and then we will perhaps together act more ethically too.
Yeah, and for me that brings to the question of time and temporality, and I thought your bringing in of this, I'm not familiar with this Deleuzian concept, and I think that it's wonderful. So thank you for that. I've done work around the aesthetics of care in the past, and temporality is so vital to it. But what I'm finding is that it has to be a non-chronological temporality, because if you're talking about more non-human organisms and non-human and even metabolisms, they function at all different scales and at all different temporalities. And so it's not, yeah, I think I'm not sure that Edelman is a, or even here nows are useful thinkings around this. My understanding is that the queer futurities idea is from Munoz, and he talks about it as sort of a utopic horizon that we perform towards this idea, and it's through the excess of the performance and the aesthetic that we create some hope or some movement towards it or around it, probably more around it. And it's a collective thing. And so if we're talking about collective or social time, we're always talking about a non-chronological time, I think. Microbes are interesting for me in terms of time and death as well, because most of them replicate themselves asexually, and so they copy themselves. So it's almost impossible to tell where one ends and begins. And so there is no kind of linearity in that sense. So yeah, I'm still figuring that out. And then if we bring metabolism and olfaction into it, into that sort of endlessness, I suppose. Yeah, I'm still grappling with the olfaction stuff, and I'm still not quite sure how Candida smells yet.
Okay.
I know how I smell. I know what I smell when I work with it, but I don't know what it smells yet.
Okay.
Yeah.
Okay.
That's part of the experimental work that I'm going to be doing in the next couple of years. I love this idea of deodorization, and I hadn't thought about it, but just yeah. For me, it's part of either the microbiopolitics, of course, and the control of microbial production, I suppose, and microbial bodies and microbial communication and reproduction.
And can I just intervene?
Yes.
Tarsh, would you mind actually kind of telling us a little bit more? Because of course, I mean, like, you both guys actually kind of, read each other's sort of like comments and papers. And I think that everyone in the room is a bit like, so I think it's good to kind of have a bit of a background on that too. So apologies for your interjection.
No. Okay, so about what specifically, Marietta?
I mean, just give a kind of a bit of a background to the concept of deodorization, like how you introduce it in your work, and what was actually the argument that Nina was kind of commenting on, so to speak.
Well, in response to that, I don't mention deodorization at all.
Okay, right.
So
right.
Nina's
this is good to know. This is good to
yeah. So Nina's prompting me to think about it.
I know where you do it in the I can it's in number 21 in the manuscript you sent me.
Sorry about this kind of like digging details, but I think that this is exciting. I mean, at least like thinking selfishly, it's very exciting for me, this whole detail. So I would like to kind of know a little bit more.
Yeah.
But I think that because precisely our seminar participants also don't have that background.
Yeah.
I think it could be interesting to them too.
I just say, I mean, you're right. I mean, just one sentence, which I really think was, I mean, now I quote you, Tarsh. The revolution in civic cleanliness has been accompanied by a revolution in personal cleanliness.
Yes.
And this came with the drive for deodorization. That's a quote from you. So you were the one who actually invented the word deodorization.
No, not at all. I think actually that's from Latour.
Okay.
Yeah. because he talks about, maybe I can't remember.
Okay.
So,
but still, I do think that the concept is interesting.
Yes, yes, and it also, okay. So deodorization would then be the process of removing unpleasant odors. I mean, it could actually be the process of moving all odors, potentially, right? It's interesting for me to think about that. And thank you so much, Nina. That's a really powerful thing to pull out from. So I really appreciate. When I came to Umeå University and the Institute of Design, which is where I'm now based, there was this big sign up at the front of the door saying, this is a scent-free zone. Please do not use perfumes, etc., etc. We don't have this in Australia, these signs up. And I love it because I don't actually like smells so much, ironically. But what it has me thinking about is how that sign tells us to police our smells and our behaviors associated with smells, and then how we not only then do a Foucauldian self-regulation of our own bodies and then the organisms that produce the smells from our bodies, but then we also are controlling, or I guess there's becomes a power interaction between cultures as well. So cultures where smells are very important in their food or in their personal hygiene or whatever it is, then that becomes a mechanism of control of that as well, and potentially colonization continues on through that sort of space as well, which I think is really interesting in terms of human culture. But we are also then creating it in the microbial culture as well, because we are then affecting the evolution and the communication within microbial systems as well. Does that answer the question? Amazing. Yeah, lots more questions, and I love the beaver. The beaver, it's wonderful, and that they could actually have an effect, I think is really important. I think that one of the reasons, I know we should take a break, but one of the reasons that I've focused on the here now is because of the urgency. And it's one way to help me try and think about the dying that's happening now and how to be responsible for it now, rather than to put it off into a future space, I think. Yeah. Should we take a break?
Yes, my dears. I mean, I would love to thank you both for a super exciting, a kind of very, let's say, delicious, and beautifully smelly dialogue. If we can think about that, like kind of a figuration of a smell, of a very nice smell that, gives us pleasure. This was actually the kind of enactment. But I mean, I would love to thank you for this. And of course, now we'll have, 10-minute break. So we'll reconvene 2:35 Central European summertime. Still summertime, the zone we are in. So in 10 minutes we reconvene, and I also right now will stop the recording so we can kind of, in a more relaxed manner, everyone can take part in the discussion. So yes, looking forward to that. Thank you all, and please, we are back 2:35. Thank you. So you can take, relax a little bit.